That's my shirt.
Yep.
Okay, so
Cut
the
that off.
presentation is all ugly and boring.
Put that on green. And
because-
I don't think so.
Let me, Daddy, don't.
Okay.
It's ugly and boring because I wanted a full presentation, but when you present research, they make you use this template. They got all the way and stuff on it. So, anybody know what a Nobel Prize is? What's a Nobel Prize? What's a Nobel Prize?
A Nobel Prize is, let's say, you did something for a mayor and you got like an award. That's a Nobel Prize from the mayor.
Can I say, I thought it was pronounced Nobel.
Nobel.
Nobel Peace Prize.
Well, it's a bunch of different categories
Oh.
for the Nobel
My fault.
Prize.
That's
I don't know
right.
how it's pronounced, but
No, they gave Martin Luther King the Peace Prize because he kept peace. So you're right, it's a Nobel
it's
Prize.
a bunch of different types. But for this one, the one that I'm presenting is for physiology and medicine, I believe, if I'm not mistaken. And so there's always, different categories of Nobel Prizes. And Nobel Prize is like the highest award that someone receives if they're able to invent something or create something that changes like the population or for the good, or changes something for the people for the good. Yeah.
Does it come with a check?
Sometimes.
I heard they get $1 million.
I don't know.
It's $1,000 or $1 million.
It's $1 million. That's y'all gonna look it up. It's $1 million. So you're probably
So,
get $1 million.
what I'm presenting to you guys is the Nobel Prize laureates. Laureates is what they call them when they win the prizes. 2023, which is this year. Their names are Dr. Caitlin Carrico and Dr. Drew Weissman. And what they are awarded for is their help for the development of the mRNA vaccine production.
Say what again?
Daddy. What they are being awarded for is with their help towards the development towards the mRNA vaccine production. And I'll explain later. So their research that they were able to do was able to help the development of the mRNA vaccine, so they were able to quickly produce it when we were all going through the whole COVID pandemic of coronavirus. So you all know the virus, I think the pandemic started in 2020. Thank you. And then now it's 2023 and we already have different types of vaccines that we're able to get that is supposed to help us boost our immune system. So that if we are contracted with the virus, then we're able to fight it off. So, with this outline, I'll talk to you guys about who these people are, the vaccines before the pandemic, meaning before don't cut me off because I get to losing my mind, losing my train of thought. But what their research, before their research were able to help how the vaccines were produced. So we'll talk about that, and then we'll talk about how vaccines work today. We'll talk about the immune responses, so what your body does when you contract a virus or a bacterium. And then their inspiration behind why they wanted to revamp the mRNA vaccines. We'll talk about their collaboration, dendritic cells. I'll explain to you guys what those are. The discovery concerning nucleoside-based modifications. We may not get into that because that's pretty deep. And then we'll talk about their publication, which is the research. Once a scientist produces research, then they have to write a paper on it. The paper has to be, it goes through an internal review board, and then that has to be, what's the word? It has to be okayed, for lack of a better term. And once they say it's okay, then the research can be published, and then it'll be pushed out for the public to read. So we'll talk about that 2005 publication, which is the reason why they got the award. And then we'll look at a little bit of data. It gets a little meaty, so just a little bit of data, and then we'll go through some conclusions. So, Dr. Caitlin Carrico was born in Hungary. She was born in 1955, and she went to Szeged University, which we won't know anything about that because it's in Hungary. And she earned her PhD in 1982. She did a postdoc, which is something that scientists do after they earn their PhD, so they become a doctor and then they do research for another university. She did her postdoc at the Hungarian Academy of Sciences until 1985, And then she also went to Temple University, and then she became an assistant professor, which is, if someone earns their PhD, then they're also able to teach at a university or a college, is what a university is, as well as they're able to lead research. And so that's why she was an assistant professor, and she was that at University of Pennsylvania until 2013. That was where she met Drew Weissman, who was born in Massachusetts in 1959. He earned his MD, PhD from Boston University in 1987. He did some clinical training at Harvard, and then he went to medical school. He did some postdoc research as well as at the National Institute of Health, and then he also started a research group at University of Pennsylvania. So that's where they met, at the University of Pennsylvania. So first we're going to talk about vaccines before the pandemic and how they used to work.
Can I help you?
So first we'll talk about vaccines before the pandemic and how they used to work. So vaccines, of course, are an injection that you get inside of your body. So I know you guys probably hate needles, because I hate needles. But if you get a needle injected into your body, then they have a vaccine or a chemical inside of there that is supposed to give your body a head start for immune responses. So if you are able to contract the virus, then your body already has a head start to be able to fight off the virus. And so the way that they made vaccines before the pandemic is they used a specific virus that is either killed or weakened, and so it's not as strong as the virus will be, and they're able to mix it together with recombinant proteins as well as a viral vector, and those are just specific things that help the RNA or the DNA to build off what it needs to build. So they combine those together and then they put it inside of a vaccine, and once your body contracts it, then you already have the specific machinery to be able to fight off whatever it's targeted to. So if the vaccine is for COVID, then once the vaccine is put in your body, if you are contracting COVID, then your body already is able to fight off COVID. Or for stuff like polio, measles, yellow fever. So limitations. With this specific way of producing vaccines, they had to do cell culture, which is when you take some cells and you grow them and you treat them with the medicine to make sure that it does what it's supposed to do, and then you use that to make the vaccine. So it takes a really long time, so they're not able to produce quick vaccines very quickly. Like, once again, it's 2023, the pandemic started in 2020, we already have multiple vaccines. So that was the biggest limitation. Also, another limitation was, I forget. It's a limitation, it's not important.
Limitations, right?
It's not important.
That's the limitation for the vaccine, so it won't work, or?
No, it works. It just takes a long time to get produced. And it needed to be quicker with the pandemic, because we lost so many people with the pandemic that we needed something quick to be able to stop people from dying or stop people from getting extremely sick. So this is how vaccine works. We already talked about that. If a bacteria or virus goes inside of your body, then it multiplies, and once it multiplies, then your body then has different cells like T cells or B cells, which are immune cells that fight off the virus, and then you're back healthy again. And so this is how the injections work with vaccines. They inject you with it, and what it is something that's supposed to mimic the virus. So it's not exactly the virus, but it mimics the virus. Once that is injected, then of course that multiplies as well, so that you have the same machinery that you would if you contracted the virus. And then once you are, say you get sick with COVID, I'm just using that as an example, then they're able to have stuff like antibodies, which looks like that, a little Y figure, that is able to come in and fight off the virus. So, immune responses. Immune responses are carried out by white blood cells known as lymphocytes. And there are two specific types of immune responses. You have antibody responses, and then you have cell-mediated immune responses. And those are carried out by, like I said earlier, B cells or T cells, which are lymphocytes or white blood cells. So you guys know you have white blood cells, you have red blood cells. Neutrophils, monocytes, and also macrophages. You guys can ask like a million questions after if you want to. So, this is the Dr. Carrico and Dr. Weissman. This is their inspiration behind the exploration of mRNA. So mRNA is messenger RNA. How can I break this down really quickly? Okay, so you have the central dogma of biology. First, you start off with DNA, and then DNA is transcribed into RNA, so it just translates it into RNA. RNA can have something called messenger RNA, which translates it into protein. So if your body has a specific sequence, let's just say a DNA sequence, like I don't want to go too deep. If your body has a specific sequence for COVID, and then it starts producing off the messenger RNA, then it is telling your cells to start producing the protein for that specific sequence. Does that make sense at all? Once again, our body it creates genetic information, which is transcribed into mRNA, which then gives protein. So,
Go ahead.
and
So we got all
so,
this traffic.
yeah, no. And so the science back in the day, in 1980, they came up with in vitro transcription, which is just a different technique that they use to create mRNA vaccines.
In vitro what?
In vitro transcription, which I'm not going to go into this today. But just know that's a different type. You want the presentation to be 30 minutes and not 4 hours. So I'm only going to go into the things that are important to me right now. So in vitro transcription is just, remember, that's just a type of way that they produce mRNA vaccines. And then that's when mRNA vaccines took off. But of course there were still roadblocks that remain, which were the fact that the mRNA that's created is unstable, so it wasn't able to stay alive in the body long enough, or it may have off-target binding, and then it was challenging to deliver, and so it was hard to get it into the body without it doing different stuff that it didn't want it to do. And then you carry lipid proteins, which encapsulate or it covers the mRNA to help it travel to the body. So that's like a longer, harder process to do, as well as it gave off an unwanted inflammatory response. So inflammation is that swelling that you have in your body, and if they produce the mRNA vaccines, then it creates swelling, and of course we don't want that with the vaccines. So that was the reason why they wanted to look into mRNA vaccination. So this is their reasoning behind their collaboration. So Dr. Carrico, she studied RNA modifications, I believe, and what she wanted to do was produce the mRNA vaccines, but was to get recognition for the mRNA vaccines or used as a therapeutic, but she couldn't do it because of all the limitations. So she was like going through a lot, losing funding. She got demoted in her university. And a lot of people just looked down on her because they, of course, didn't believe that they could get this research done. Now, Dr. Weissman, he looked at dendritic cell function and immune surveillance and the activation of vaccine-induced immune responses. So, dendritic cells are found everywhere, and they're responsible for the initiation of adaptive immune responses. So they are the reason why our body has the immune responses that it has, like, say for instance, inflammation. So how they work is they look like this little circle with the little arms, and if say bacteria comes inside, then they have toe-like receptors, which are these little hook things on them that grab the bacteria, they recognize the bacteria, and then they push out something called cytokines. And cytokines are what tells the body to produce what it needs to produce in order to come up with stuff like inflammation, or to be able to produce antibodies. Okay, that was a lot. So with that being said, they came up with trying to research how different RNA types will interact with the immune system. So changing RNA to figure out what it will do to the immune system. So what they discovered is that those dendritic cells that recognize bacteria and then produce cytokines, they recognize that they learned that dendritic cells recognize in vitro transcribed RNA as foreign. And so if the body recognizes something as foreign, then what it does is it induces an inflammatory response, for instance. And that was what they were talking about back then with one of the limitations as to why they wanted to study mRNA vaccines. The fact that it gave off that inflammatory response that was unwanted. And so with in vitro transcribed RNA, once again, is a technique that they use in lab to produce mRNA. So it's not real mRNA, but they use it to make it. And then nucleosides in mammalian cells, which is what we have, is taking real DNA, real RNA. Nucleosides are, you ain't got the whiteboard in here no more? You ain't got the whiteboard in here no more?
No, it's right there.
Oh, well, that's a lot. Nucleosides, so you know DNA has adenine, it has guanine, it has thymine, and it has uracil, whereas RNA has adenine, guanine, cytosine, and uracil. No, cytosine. Okay, I'm losing my train of thought here. AGCT. AGUT. Oh, that's okay. I don't need it.
Okay.
I just figure it'll keep them
Okay, well.
connected. But anyway, that's okay. Thank you.
I'm just biting my lip not to ask for questions.
You can ask questions. I just don't want the presentation to be 3 hours long. So nucleosides in mammalian cells, so RNA has U. Oh, it's so hard to explain. Like, okay. So you guys see how DNA is like the
Helix.
the helix.
Double helix.
Double helix. Inside of DNA is those specific bases. So let's just say the yellow one is A, and then A binds to A, the adenine, to guanine, cytosine, and thymine. So A binds to G, which is green. And then DNA has thymine. No. GUAT. Ooh, I'm losing my train of thought. That's terrible. I need to go back over that. But with RNA, instead of it having T, I believe it has U. So it has uracil. So those are the specific bases that makes DNA do what it's supposed to do. It binds together, and then it has that helix shape. So those are the bases. So what they noticed was that when they had in vitro transcribed RNA, it's unmodified, so it looks like normal uracil, uridine. Whereas if they have sorry guys. Whereas if they have nucleosides from mammalian cells, which is human cells, for instance, then it's modified, which means it's changing in the body. So the fact that it changes in the body, it allows it to go, once it is introduced to the cell, it allows it to go through without giving that inflammatory response. So the body doesn't want to fight it off. Whereas once they modify it, then of course the body fights it off. I'm sorry, once they in vitro transcribe it, then the body fights it off. Yeah. So that is where they come up with the 2005 publication, which when scientists produce research, then they have that publication or that paper that comes with the research that includes data and background information. They publish it into specific journals, and then journals have like a different level, like immunity may be higher than Cell journal. So what they did is they produced it in the Immunity Journal in 2005. So that was 15 years before the pandemic started. It was when they published their research. And the title of their paper is The Suppression of RNA Recognition by Toll-like Receptors, which is those little hook things that were on dendritic cells. The impact of nucleoside modifications, which is that you being modified in mammalian cells or human cells, would you say, versus it being unmodified in in vitro transcribed RNA. And then the evolution of the origin of RNA. So their aims, what they wanted to look at, or specific questions, is will nucleoside modifications have the ability to suppress the immune stimulatory effect of RNA. So will those specific modifications be able to stop the inflammatory response? And then they looked at, well, different nucleoside modifications react differently with toll-like receptors. And so if they modify or change the uridine to have like a methylated group, will it be different than giving it something like what they call pseudouridine? So like, that's like saying if I decide to polish my nails brown on this hand, but blue on this hand, if I put it in front of a light, will it change that light differently depending on the different colors? So that's what they mean by different modifications. And then what is the relationship between a specific amount of modified nucleosides versus the suppression of immune stimulation? So they also wanted to look at if they have more modifications, will it suppress, or suppress means to minimize or to make it smaller, will that suppress the immune stimulation? So will it make the inflammation smaller and smaller and smaller? So this is some of the data. Once again, it's really meaty, so I'm just gonna sum it up as quickly as I can.
We got 5 minutes.
Okay. Trying to see which one. I'm just gonna present one of their graphs.
We have 21 minutes.
Okay, maybe this one will work. Okay, so remember one of their questions was, will the relationship of immune stimulation be what they call inversely or directly proportional? Those are specific relationships that scientists look at. Like if something is increased, will it increase something else? Or if something is decreased, will it decrease something else? Or if something is increased, will it decrease something? So they were looking at that specific relationship. So what this is showing is those RNA sequences that I was telling you about. So remember how RNA has U's, A's, G's, and C's? Yeah, and C's. So the first one is, all of this is in vitro transcribed RNA, I believe. And so the first one is their control. When you're creating experiments, you always want to control something that you can look at and say, and compare the rest of your data to. So they gave a sequence that doesn't have any modifications in it. And then the next one had a modification of I forget what UM is. It's like 2' something really long. So that's one modification. And then they looked at another one, which is M5C, which is another modification. And they looked at another one, which is pseudouridine, which is another modification. So those 3 have modifications. And then, I'm sorry, ORN is what they call oligoribonucleotides. And so when they say oligo, you're so silly. When they say oligo, that's how you know that is something that is chemically synthesized or something that's made. That's why they call it in vitro transcription. And so 5 and 6 are, wait a minute. These are chemically synthesized, these are in vitro transcribed. Once again, the in vitro transcription has no modification on the U's. So you can see like, after this C is U, but instead of having a U on this sequence, you have UM as a modification. Or right here, AUCC, PUGGAUCC. This one is a modification on a C instead of a U. That's interesting. BUGGAUCCGGCUU. This one is another modification on the U. Okay, so, and then this is more data that's showing TNF alpha, which TNF alpha is called tumor nectar, which is a cytokine. So remember, you have those cute little dendritic cells, which are circular, have these little things hanging off. Bacteria is grabbed by the toll-like receptors, which is that little hook thing, and then it produces cytokine. So one cytokine is TNF alpha, and TNF alpha I think it's also an inflammatory response, immune cell, or immune cytokine. Okay, I'm almost there. And so what they looked at is lipofectin, which is something that they put cells in, and you can see lipofectin is a low amount of TNF alpha. So on the y-axis is showing the amount of TNF alpha, and then on the x-axis is showing like the different modifications or just lipofectin alone. This is their positive control, which is showing if they do have an increase in TNF alpha, this is what something would look like. This is that control, which is the one with the no modifications. You can see that is much higher than the ones that have modifications. So it's showing the unmodified RNA is what induces the inflammatory response. And then one last thing I want to show you guys is this data down here, which is in gel. So what they do is they take two chemicals, a solid or something like a powder, and then the liquid, and they mix it together and they heat it up and they pour it on this plate. And once it's on this plate, then it becomes like this gel, like a jelly type of thing. And what they're able to do is they're able to inject each one of these things. So this is TNF alpha, and it's showing like the amount. Once the bigger the signal is, or the darker something is, that's showing that is more in there. So they're able to inject each thing in there, like the medium, or the poly, or the R848, which is another positive control because it activates TNF. It activates 7 and 8, or something, I forget. Yeah, so I'm sorry, I'm almost there. So these last two are the in vitro transcribed RNA, and as you can see, the signal is how they interpret this data. The signal is much higher than the ones that are modified, so that's, once again, showing the accuracy of their data. Sorry, I'm still gonna have to use my iPad. Okay, so some conclusions. What they were able to discover is, or what they were able to do is to help with the very fast production of mRNA vaccination, because the type of, the way that they're able to produce mRNA vaccines is much quicker than how they used to do it back in the day. So they don't have to do the whole cell culture process, because they learned that nucleoside modifications are able to suppress the immune stimulation of effective RNA. So what they do is they just go in and they modify the mRNA that they make so that it doesn't induce that inflammatory response. The rest of this stuff isn't as important. Those are my citations, and I'm done. Any questions?
Good.
Yes.